few housekeeping issues. As everyone is doing, please keep your microphone off during the presentation. We all have such Zoom fatigue. We recognize that. So please don't feel pressured to have your camera on during the presentation. However, when we're in the breakout rooms afterwards, we ask that everyone have their cameras on and ideally put your screen into gallery mode so you can see everyone. Please be aware that the talk and the question and answer period will be recorded, but not the breakout sessions. So if there are things, if you do not wish to have any questions recorded, please type your questions for the Q&A session in the chat box and to me, and I'll ask it for you. If you prefer for the question to be anonymous, please directly message me and I would be very happy to ask on your behalf. If you choose to talk about this wonderful workshop on social media, we ask that you tag us because we would love to hear from you, and please feel free to follow us, and here are our social media handles. So just a quick run through for the agenda. I think everybody has this in their packages, so obviously we're at the beginning. This is Mountain Daylight Time. I recognize people are across time zones, so just adjust accordingly. I will, starting in a few minutes, formally introduce our keynote speaker, Dr. Rebecca Shansky, and truly delighted to have her here today. After Dr. Shansky's talk and the Q&A, we'll move into our breakout sessions. So you will, and Dr. Drogo's Lauren will explain that more fully later. So thank you for that, Lauren. But just so you know, you will automatically, magically disappear into your breakout room, where you'll have the opportunity to meet with one of our outstanding and truly internationally recognized mentors for their work in sex and gender, and talk about how you can incorporate these parameters into your study design. So we want people to leave with the tools that they need so that you can apply these things into your research when you go back to your home institution. We ask that within each of the breakout rooms, there is a note taker who will record on paper, at least, the discussion, who will then, when we go back to the large group discussion, which will be moderated by my colleague, Dr. Thompson, just to report back, like, what did your group talk about? What were the big obstacles, the big opportunities that you came up with? Then we'll take a 10-minute break, move back to our breakout session number 2, where we'll talk about incorporation of sex and gender into data analysis and reporting. We do ask that the note take, and then we'll come back to our large group discussion. We do ask that the note takers provide their notes to Lauren at the end of the session, because we'd like to hear and record about what types of things came up in your work. What are the big obstacles? How can we help? And then we'll have our wrap up and closing. So in terms of conflicts of interest, the Scientific Planning Committee, we've reviewed all the conflicts of interest from all speakers, mentors, and moderators, and nobody has any that are could impact this workshop. So please recognize that the work that and the skills that are being shared today come without any concerns that way. I really want to emphasize that the mentors, we are very grateful for their time and expertise, and it's truly going to be a fantastic day. So without further ado, and I'm sorry, there we go, my computer has just died. So sorry, Dr. Shansky, I have your, I'm actually going to pull this up. My very much apologies. My second device has just, there we go, died. All right, so Dr. Shansky, we are delighted to have you join us today from Northeastern University in Boston, Massachusetts. Dr. Rebecca Shansky earned her PhD in neurobiology at Yale University and completed postdoctoral work at the Mount Sinai School of Medicine before joining the faculty in the psychology department at Northeastern University in 2011. She directs the Laboratory of Neuroanatomy and Behavior, where her team uses rodent models to explore the links between brain structure and function, focusing on how individual differences in response to trauma shape long-term memories. She's a vocal advocate for gender and sex equity in experimental design, highlighted recently in her Science magazine perspective piece, Are Hormones a female problem for animal research. And we will circulate that paper at the end of the symposium, because it truly is an outstanding piece, as is all of Dr. Shansky's work. And I am personally a longstanding fan. So thank you very much for joining us, Dr. Shansky. And I will please start sharing your slides now, and start when you're ready. Thank you.
Okay, thank you so much, Sophia, for such a flattering introduction. I am really excited to be here. Hopefully everyone can see my slides okay. And so I think part of the reason that a neuroscientist, I'm here at a cardiovascular focused program is mostly just because I have been the noisiest when it comes to talking about using males and females animals in preclinical biomedical research. And so the science paper was the first one, and the second one came out earlier this year in Nature Neuroscience, in which I break down a lot more beyond, but I'm going to try and kind of sum up some of the major things that I have been thinking about and help you think about it for your own research. And I am just really, really excited for the conversations we're going to have today. So I wanted to start with a figure from what I think is now probably one of the most canonical papers on the use of, or sort of the imbalance between male and female animals in biomedical research. And so this is Beery and Zucker from 2011. And essentially what they did is they went through a century's worth of scientific research papers and just asked, did the authors of these papers use male or female or both animals? And the results of that are plotted here by year, and you can see they start down around the turn of the century, and then work their way up to 2009, which is where they stopped for this paper that now is 10 years old. But what you can see is that this gray dotted line here are the papers where they just didn't say whether the animals were male or female. And every other line here, the black is animals that were, or papers that used animals of both sexes. The brown are papers that used just female animals, and the turquoise are the papers that used only male animals. And what I think is pretty striking here is that by and large, it was just not common practice to even bother saying what sex the animals were. It was not a consideration at all. And what you can see is that somewhere around in the 1970s, that practice became less common, and the sex of the animal was indeed noted in the methods section. But what you can see is that there's sort of a crossover here where suddenly we have a lot more male only papers, whereas the both sexes papers and the female only papers basically stay the same. And so what this suggests to me is that all along we were actually seeing, you know, when it wasn't specified, we were seeing research that had been done only in males. The authors just weren't telling you that. And, you know, to me, what that says is that the males were considered such the obvious default for understanding organismal physiology that it didn't even need to be said. It was so, you know, just sort of a given that we would study male animals in order to understand you know, the brain and the body and how everything works. And so this, you know, you can see these numbers really have not changed, especially the both sexes. Things may be creeping up a little bit, but not nearly as much as we would like them to. So one of the things that I feel like data like this sort of promote is what is with this big imbalance? Why don't scientists want to study females? And I'm sure you can guess the answer. It is hormones, because females have crazy hormones that change every day. And so in these graphs, you can see on the left, the hormonal levels during the rodent estrus cycle, which is about 4 to 5 days, and the human menstrual cycle, which is of course about 28 days. And in both cases, you can see that they are characterized by sort of isolated peaks of estradiol, progesterone, luteinizing hormone, and follicular stimulating hormone. And the general idea was that this would make your data too complicated. What if you had one animal in proestrus and one animal in estrus? How could you possibly understand anything about animals? And this is of course faulty logic here. But this was kind of the general thinking, even if it wasn't usually said, put in writing by the authors themselves. However, there was one article where this was sort of stated explicitly. So this is a paper from Science in 2010, essentially just explaining why researchers don't want to study females. And this heading, "The Trouble with Females," was literally in the paper, which I just think is so crazy. Like, this is, my lifetime as a scientist. And they are basically arguing that it's just easier to work with males because of the females. And here, this researcher just said, basically, the data are uninterpretable. So nothing you collect from a female animal, if you don't know their hormonal status, is useful information, which I just think is so crazy. And they go on to say that even if you have it's going to be really messy data. And so this was kind of the party line for a century, essentially. It reminded me of this kind of meme, which is this idea that men are very simple and women are very complicated, and how could you possibly understand anything about women or female mice because of these hormones. And so this was seen as such a reasonable assumption that nobody ever actually asked it empirically until a little bit less than 10 years ago. And so this was done for both mice and rats. And essentially what these researchers did was they performed meta-analyses of hundreds of papers where they looked at the variability in the data, right? So this is what scientists have been afraid of when it comes to the estrus cycle, is the estrus cycle is going to make my data more variable. Obviously, as scientists, we want really clear, clean data that are not variable. And the assumption was that female data would be more variable than data that you collect from males. And it turns out that is not the case at all. In both of these studies, they found that male and female, or data collected from male and female mice or rats, were equally variable. And in some cases, the data from the males was even more variable than the females. So now we can say, all right, this assumption that the estrus cycle is going to mess everything up and make your data uninterpretable is not true. And so now how can we kind of move forward from this? Okay. And so here's another little secret. Males have hormones too, and they actually vary a lot. So hormones like testosterone can fluctuate throughout a single day. And then from animal to animal, if you've got your mice in a cage and they're going to establish a hierarchy, a social dominance hierarchy, your alpha male could have as much as 10 times more testosterone than your subordinate males. And so this is something that we've known since the 80s. It, of course, has never been a cause for concern or hand-wringing when it comes to the variability that these kinds of naturalistic influences might have on your scientific data. And So there's clearly a double standard here in terms of what we consider acceptable variability, acceptable causes for variability in our data between animals of each sex. And so I wanted to know where this idea came from. Why do we associate females and women with hormones, and we don't really have that same kind of sort of like natural association when it comes to thinking about men? And it turns out, if you go back in some of the sort of older gender studies literature, we can blame the Victorian era biologists. So up until that point, the sort of message or the narrative from biologists was that women were essentially an inferior version of men, and that fell kind of out of fashion around the Victorian era. And so the scientists said, well, okay, so we've got to come up with like a new theory on how how men and women are different from each other. And but we can't say they're inferior. So what if we just said, okay, you're not inferior, ladies, you're just different. And we're going to give you your own set of qualities, and here's what you are being assigned. Sensitivity, emotionality, instability, and these things are due biologically to the fact that you have reproductive hormones. And so this is where it came out. It came from scientists. And that message, that narrative has really stuck in society. And the goal here for the scientists was that, okay, women are not inferior, but they are different, and they are different in ways that make them unfit to lead society, and therefore the patriarchy is preserved. And so I think, we still see ideas like this today. This is something that has really kind of woefully persisted in the way that we think about how men and women are different from each other. And so this is something that I think, unfortunately has also infiltrated the way that we think about male and female mice and rats in the laboratory in terms of how valuable they are to basic science research and what they can tell us about biological processes. And so sort of the 5 or 6 years ago, both the NIH in the US and CHR in Canada introduced mandates for their grant applicants. And so in the NIH, it's called Considering Sex as a Biological Variable, or SABV. And for CHR, it is the Sex and Gender-Based Analysis, or SGBA. And they're very similar mandates, essentially saying that you, as a researcher who's applying for funding, need to consider the role that sex plays in your research. And essentially it said, you need to use both sexes. And so the sort of collective cry went back exactly where we would expect it to have gone. What are we going to do about the hormones? So, what this indicated to me is that once again, now researchers who had been only studying males forever suddenly were like, oh my God, I have to study a female. How am I going to learn anything with all these crazy hormones? And so in the past, if you wanted to study females, or if you wanted to study sex differences, this was kind of a common thing that you needed to, it was like great that you wanted to study sex differences, but you needed to figure out how to deal with those crazy hormones. And this is true for me too. So I started doing sex differences research basically my first day of grad school, and it was the immediate thing that we needed to figure out is what are you going to do about this crazy cycle? And so there's basically two main ways to do it. So one, you can track the estrous cycle, which is sort of a proxy for hormone level, how much estradiol or progesterone the animals have. And you can maybe study animals that are in one or two different phases of the estrous cycle and compare and see whether your data are different based on that. Or you can essentially remove circulating estrogen altogether by removing the ovaries. And if you want to see what happens if you put the estradiol back in, then that, is also an interesting experiment to do. But these were the decisions that most people had been making if they wanted to study females, especially in my field. And I think that if you have experimental questions or hypotheses that are centered on the role of ovarian hormones, then these are reasonable steps to work into your experimental design. There is a whole field called endocrinology, which is specifically asks questions about how hormones affect physiology, neurobiology, things like that. However, these mandates do not require assessment of the influence of hormones on your data. This is not the goal of SABV. And they do not even require statistical power to detect sex differences. So really all they're asking is that you use equal numbers of males and females, and this is what can help inform us of whether sex may play a role in your data. And so what this means from an experimental design standpoint, if you've never studied both sexes before, is you can just use the same number of animals in your experimental cohorts and just have like a mixed group of males and females. Now, what happens when you get your data? Well, you have to look at it, believe it or not. And so there's a couple of different ways things could shake out. If you have a study that is mixed cohorts of males and females, but you're not really powered to detect sex differences, you've got to look at your data and make kind of an educated guess. So if this is what your data look like, right, here's your y-axis, here's your group. The data from your males and females are essentially completely intermingled. The variability is the same. Everything is the same. You probably don't have a sex difference in this particular measure. And so in that case, it is great. You can keep using mixed cohorts and always keep looking at your data. If it turns out that again, you're not really powered to detect a sex difference, but if it seems like the males and females are separating or stratifying out according to sex, then you have a decision to make. So here you may have a sex difference in your experimental metric. What I would do in this case is add more animals, increase your sample size so that you are powered to detect whether or not there are sex differences, and then you may want to move forward examining the males and females separately, kind of in parallel. And you can look for points of both convergence, right? So maybe the next experiment you do, everybody kind of like comes back together again, depending on your manipulation, or further divergence. So just look at your data, use the data to help you make decisions. This is what science is. Now, you may say, oh, but if I have to do that, if I have to add more animals, that's going to be really expensive and it will keep me from investigating the mechanism of my area of research. And, I feel your pain. But remember, first of all, that even before the mandates, there were a lot of us who were doing these kinds of studies designed this way with the same amount of money that everybody else had. The other thing I want to remind you, which was pointed out by a reviewer I had recently, sex is a mechanism. And so just imagine you're going through your research and you unexpectedly found something else that was influencing your data. You would want to follow up on that, right? Finding sex differences is interesting and cool and something that really gives you some information about the biological factors that do influence your data. So try and keep that frame of mind, I think, and then you won't be crying Dawson over here. Okay, so a couple of other little myths I want to address. So in that first scenario where you didn't have any sex differences, a lot of people are saying, oh, well, there's no sex differences, so I'm just going to go back to using males. This is not okay, because sometimes even when the one metric that you happen to be looking at, the males and females end up kind of in the same place, there still may be underlying sex dependent mechanisms that are getting them to that same place. This is what Catherine Woolley has called latent sex differences. And so you always want to keep going with those mixed cohorts because you never know when things are going to essentially pull apart. So it's really important to keep using both sexes, even when you don't have a sex difference. The other thing I want you to think about for a minute is, why is that instinct to return to one sex? What you're essentially saying is it is easier to study one sex than it is to study both sexes. And maybe that's true, but you're kind of implying within that desire that you think there may be sex differences down the road eventually, right? Like at some point, stuff's going to get kind of messy, and then you're going to have to deal with it, and you don't want to deal with it. So again, think about where these sort of responses come from. If there's no sex differences, then there's no sex differences, and you should be able to keep going with mixed cohorts forever. And then the data that you put out into the world is representative of both sexes. The other thing I hear a lot is, well, I started this project in males, so I'm just gonna finish it in males, and then I will move on and start looking at females armed with the information I have from males. And so this is not really okay, because what ends up happening is the females become like a side project, or sort of a follow-up study that isn't going to get into the same impact journal that your male study is, because it's essentially a replication study. And that means that when people are looking for information on your topic, they're going to look at your male study and say, oh, this is what's true. Even if what you ended up finding in females was totally different, it's not going to get the same recognition as being true, real scientific knowledge. It's going to be like a female thing. This is just, this is all comes back to some of these societal, kind of unconscious biases that we have. And what we want is to get to a place where we view an experiment that's done in female animals as equally valuable to scientific knowledge as an experiment done in males. It kind of perpetuates this idea that whatever you study in males is like true and real, and whatever you study in females is like, how are they different from the males, right? So how are females deviating from the male data? And I think that that's a very dangerous kind of framing to see the way we put scientific knowledge out into the world. The other thing is, when are you really finished with a project? You can always sort of say forever, I just going to do this like one more mechanistic study, and then I'll do the females, I promise. It's never going to happen. So start with males and females, and always keep going with males and females. And so one thing I want to touch on is, what are the kinds of things we miss when we only study one sex, or when we design experiments in males and we don't validate them in females? And I'm going to give an example from my own work, which uses classical Pavlovian conditioning, which you've maybe learned about, but just to give you a quick catch up. So this is a study of associative learning. And so the way this works is, we put a rat in a chamber where it hears an auditory tone. And then immediately when the tone ends, it gets a little foot shock through the floor. And so it learns to associate the tone with foot shock. It expresses a fear response when it hears the tone in the future, even if it's in a new environment. And so what we normally look for when we're trying to say, how well did the animal learn this association, is a freezing response. And you basically, this is an animal doing a freezing response, and essentially you just say, how much time does the animal spend freezing? And so it's just sort of like a yes or no thing. Is it freezing? Anything that's not freezing just basically gets thrown out. But there are probably some other ways that animals might express fear. And what we found in our lab is that males and females use different behaviors to show their fear. And so I have a couple of little videos here for you. So this is a male animal, a male rat. And he just heard that tone. Don't worry, you're not supposed to hear it. And you can see he just sort of runs into the corner and now is going to engage in this freezing response. And you can see he really is completely frozen. The video is still going, but this animal is really just completely frozen. And that's basically all he does. In contrast, when we see females, we see something kind of different. So here she is, she's just exploring her environment. And I think this video shows when the tone actually comes on. So there it is. So you can see she did like a big spin. She did running into the corner. She's kind of looking around. She will do a little bit of freezing. A little there maybe. And then kind of creeps out to see, is it safe? But you can see, what I think you can appreciate here is that from the time the tone came on, that animal exhibited a lot of different behaviors that do all appear to be related to the fact that this fearful stimulus is present. And whereas the male basically just did that one freezing response. And so that animal, if we were just scoring it on freezing, it would get a really low score, and we would be like, oh, well, these animals just must not be afraid. Or even worse, they didn't learn. And clearly she knows what that tone means. And so what we're trying to do in my lab is work on better ways to capture this this sort of range of fear behaviors. And so we're doing that with collaboration with my colleague Bob Data over at the Harvard Med School, who's developed this really amazing machine learning tool that uses a 3-dimensional depth camera, like the camera you get with your Xbox, to capture the animal's behavior in 3 dimensions. So not just where is it in space, is it moving or not, but really what is it doing with its whole body. And then the machine learning tool comes in and essentially breaks down all of these different poses into what they call syllables. And then you can look for different patterns of behavior surrounding some kind of event like a conditioned stimulus. So we're really excited to take advantage of this and keep asking questions about sex differences in fear behavior. Okay, so just a couple of final thoughts here. So one thing I really hope you take home from this is that female animals are just as valuable and informative to basic science as males are. And I really mean this in a basic science way. This is not just about women's health, which of course, very, very important. But I think what we're gonna find from doing these, from increasing our use of both sexes, is that the translational value of using both sexes is not just going to be female rats tell us about women and male rats tell us about men. I think we know, first of all, gender is far more complicated than that. And I think that it doesn't have to work that way in order for the information we gain from female animals to be useful. It could translate in any direction we want it to. Hormones are not a nuisance that need eliminating. They are part of the fundamental biology of both male and female animals, and you can choose to manipulate them in order to specifically ask questions about what role they play in whatever it is you're studying, but they are not some problem that needs to go away. you don't have to be an endocrinologist to conduct research in females, right? So I think one of the basic things here is you can take a female rat and do the exact same experiment on a male rat. If you find something interesting, then you can start to ask what might be causing those sex differences, and hormones are low-hanging fruit in that respect. But you don't necessarily have to all of a sudden learn how to be an endocrinologist just because someone told you to study females all of a sudden. Speaking of, what's causing your effects, variability is interesting and it's cool, and it is really important for the goals we have when it comes to translating basic science into clinical applications, because people are really variable too. And looking at the variability instead of trying to reduce it down can actually be what better informs things like personalized medicine. So embrace the noise is my my message there. Along those same lines, I hope that you will view SABV and SGBA mandates as an opportunity to really better understand the scope of what is possible in mammalian biology, and not as something that is inconvenient or labor intensive. I think it's going to be worth whatever extra work you might end up doing for that. Okay. Finally, there are a lot of resources out there to help you practice good SABV research. These are a couple of recent papers that have been put out there. This one is especially nice because it tells you the different kinds of sex differences that you may observe, from qualitative to latent, which I mentioned, or quantitative. And they all have these great decision trees to help you make decisions about what to do next if you find a sex difference. And so these are a couple of others over here on the side. And so again, there's so much amazing information out there. There are so many great researchers who have been thinking about sex as a biological variable for longer than even I have, and I feel like I've been thinking about it a long time. So I really encourage you to take advantage of your colleagues who really have been doing the work to put this information out there for you to use. And so that is all I have to say. Thank you so much. Oops. And I look forward to our discussion today.
So thank you so much, Dr. Shansky. That was absolutely amazing, which I knew because you're such a great speaker and you have such an incredible track record in this area. So thank you for sharing that with us today. There already is a question in the chat. Feel free to open it and read it, but I'll also read it from Dr. Raj, who's one of our mentors. So just thank you for the practical approach to SABV. Using the same number of animals makes sense. However, the NIH has given guidance to reviewers that this is okay, that adequate power for detecting sex differences is not required. And this is a very real review that we have all gotten from reviewers when we submit grants. So I don't know if you have any comments on that.
Yes, so the NIH has explicitly said that addressing SABV does not require testing for sex differences, which means you don't need to power your studies to detect them. You just need to use equal numbers of males and females. So I think this, to my knowledge, has been, kind of at this point at NIH review, I think the grant writers know what to say in order to kind of like clear that hurdle. Because I know, despite, I think, and this was the case before SABV, there are instructions to reviewers and not every reviewer goes along with, they sort of put their personal spin on things. So you are still going to find reviewers who say, well, what if there are sex differences? What are they going to do? And I would encourage people who are writing the grants to consider putting in something along the lines of what I put in this talk, which is that, we're going to start with underpowered studies, but equal cohorts, and we're going to look at the data and, make an assessment about whether or not we may have a sex difference, increase the ends if it looks like there is, and, go from there, right? So doing good SABV work is not like a one and done kind of thing, right? It's a process, like being a good scientist in other aspects as well. So to me, it's the job, at least at NIH, of the SROs, who are the sort of the scientific review officers who sit in, kind of like run the study section. If somebody says they're not powered to look for sex differences, the SRO kind of has to step in and say they don't have to be at this point. Where I think things need to go in a different direction is that, like, there needs to be some accountability. Once researchers actually get the money and start doing the experiments, I've seen a lot of male-only papers come out recently from people who I know have been funded by NIH after the SABV mandate came out. And so that's where this is sort of like my new, like the next level of my soapbox, is really trying to change the culture of making people actually do it.
Yes, and I think a lot of us here would love to join you on that soapbox. So,
I've filled it out. There is room for sure.
Great. So the questions are now starting to fly in. There's a question from Dr. Norris, also one of our mentors, which is amazing, from University of Alberta. So lots of data, even in COVID, that women's cycles affect outcomes, or maybe are associated with outcomes. So her question is, should we consistently measure where the rats and/or women, and I'll say those with female biology, are in their cycle, just to account for that?
You certainly can. I think that it's an experimental design consideration like anything else. And so if you are studying something that is known to be affected by the ester cycle, then sure, it's a good idea to, continue along those lines and track the ester cycle. It's certainly not hard to do. But, I think I still worry about this kind of like knee-jerk reflex that, like, if you have a female research subject, you've got to know the hormonal status. And we have never said that about males before, even though there's, as I said, evidence that their hormones can fluctuate or change, from animal to animal even more than the females are. So I think you should make your decision based on science and not, what might be some implicit biases.
Oh, that's great. So our next question, Dr. Gwadangi, who's one of our postdocs here at the University of Calgary, who was really interested in the fear extension study and wondered if the different behavior was related to sex differences, or could it be related to dominance or social role in the cage? So things that are more what we'll call gendered factors.
So that's a super interesting question. We haven't asked that. Our animals are for rats, we only pair house them. So I think the social hierarchy is less of a concern than for mice, where you've got like 5 in a cage. We haven't really looked at that specific question. It's hard to do. Social behavioral neuroscience is like its own thing, and I don't necessarily feel qualified to know the answers to some of those things. But we are doing a lot of work in our lab to try and understand what factors might drive both sex differences in fear behavior, and then we do see some variability in both sexes within the sexes, and what might be driving that as well. So I think it's a pretty complicated question that I thankfully just got a grant to study. So now hopefully in 5 years, we'll have some more answers for you on that. But I think the social influence here is probably low, would be my guess.
Okay, great. I'm, like I said, the questions are coming in fast and furious. And please, if you don't feel comfortable, I'm getting lots of anonymous questions, so that's wonderful. So one of them is that sort of maybe we should try and meet people where they're at. So one person commented that they've heard of PIs who are worried that they will find sex differences and then they don't know what to do with it. And so, if that's the barrier, we talk a lot about vaccine hesitancy and meeting people where they're at. If the barrier is that people are worried that they will find differences and they don't know what to do with it, like, how do we overcome that? Do you have any advice?
I mean, that makes me so sad. Like, you're a scientist, like finding unexpected, like weird results is like, to me, the best thing about being a scientist. Like these differences in fear behavior were totally unexpected for us, because no one had been looking at it before. And we were like, wow, like here is this new thing. I think we kind of have to study it now. And it took a long time, and there was a lot of pushback from senior colleagues in my field who were like, this is fake, you don't know what you're talking about, this isn't a real effect, blah blah blah. And finally we did enough work where we were convinced people that this was a real thing, and now I have $1 million to study it. So I think it of course is, you would love it as a scientist if like your hypotheses were always right and nothing unexpected ever happened, but that's just not reality. And I think if you are worried about finding a sex difference that you don't know how to sort of like take the next steps on, this is again why I put some of those papers up in that second to last slide there are people who want to help you do that, and again, like reaching out to call, and I have been one of these people for a really long time, and especially as soon as the SABV mandate came out, like my inbox started blowing up because people people know that this is something that I think about. And so just like any other finding that you might have that isn't quite in your wheelhouse, you find a colleague who knows a lot more about it and you ask them, like, what can I do? Like, science is collaborative and interesting. And I like, to me, again, some of the best things about being a scientist is the ability to make connections with other people and learn about something that isn't my area of expertise. And that I'll just give you another example. We have a new finding in my lab that suggests that some of these differences, some of these sex differences we're seeing are related to the pain that the animal feels when it gets that foot shock. I don't know anything about the pain system, but I know people who know about the pain system, and I am so excited to try and to build these relationships and to learn about something new. So again, I think all of this should really be seen as an opportunity for everybody to learn how to like do new stuff.
Okay, I would have time for maybe one or two more questions. So I apologize if I don't get to your question. We've had, what impact do you feel that the absence of inclusion of female animals in preclinical science has had on clinical science, and perhaps more importantly, I'll add in clinical care?
So this is a great question. And I think there are some, there's some like canonical examples, like Ambien. Probably many people have heard of this, where Ambien was essentially only tested in males and men, and then they started giving it to women, and women's metabolism is slower, and so it was processing the drug more slowly. So you take Ambien before you go to bed at night, you expect it to be essentially out of your system by the morning. But if it's not, and you go and drive to work, that's a problem. But I think in other cases, it's almost like we don't even know what we haven't discovered, because so like, let's say you were testing a drug in a male rat and it didn't do anything. So you say, okay, this drug doesn't work. But maybe it would have worked in the female rats, and maybe this could have been a drug that specifically helped female humans. And so it's those kinds of things where we're just throwing out, or ignoring what might be possible because we're not even looking at half of our animals. So I think that's sort of, it's almost like it's impossible to quantify.
Great. So I think I'll have two more questions, but please keep the questions coming. So if we have time at the end, we'll certainly ask Dr. Shansky again for her expertise. So one of them is a question about, are there any best practices for incorporating, this is more clinical human research, non-binary sex or gender identities in human research? Oftentimes when people collect this data, how people identify, there's a small proportion identify as not cis woman, cis man. And so how do you include these people and this important data into your research?
That's a great question. I don't really feel qualified to comment on clinical research practices like that, but I do think it is important that people of all gender identities are included in the research. And there are some people I know who are specifically studying transgender people in sort of like neuroscience fields that are finding really, really interesting differences there. So I think absolutely everyone should be included. And, it's the kind of thing where again, kind of look at your data and see whether or not it seems like something is kind of pulling out there.
And sort of like you said earlier, a great opportunity to collaborate with someone who does have expertise in this area. And maybe you don't have the power to look for differences, but at least you can explore them. So that's great advice. So maybe just for our last question, prior to me passing the mic figuratively to Lauren, just to give us some housekeeping things on what's happening next with the breakout rooms. Can you give some suggestions, because it's primarily trainees as by design here, on how a trainee could talk to their PI on who might be hesitant to include SABB or SGBA, and the trainee is sees that this is affecting their particular research or study. Do you have any advice?
Yeah, so great question. And I know that, sometimes it really is going to take the trainee saying something. And I have talked to lots of trainees who said, my PI really didn't want to start studying females, but I said, I want to do this. And, I think it's really important. And I think, it might be the kind of thing where you have to say like, I want this to be my project, and I will take on the responsibility of, figuring out whatever needs to be figured out in order to, start incorporating males and females. And of course, you as the trainee, you have all these resources, you have me, you have these papers that are out there. And so I think when you come to them with the cool data that like males and females are totally different, then they're going to be all about it. So I think, it's like, of course, PIs can be very stubborn and, set in their ways. But I think the more that people speak up and say something, the more they're going to have to listen.
Great advice. So with that, thank you again. It was a fantastic presentation. Just so this recording will be made available later. So if there are some things that you missed or you wanted to go back, just know that you will be able to. So thank you again, Dr. Shansky. Everyone, please stay on. And if you could please pass, Lauren, I will pass the mic to you.
Awesome. Thank you so much. And thank you so much to both Sophia and thank you so much to Becca. That was an absolutely amazing talk. I'm going to stop